Hello friends, today we are going to learn Auxin, a phytohormone which is necessary for proper growth and development in plants. I am Dr. Alka Inamdr in Alka's Botany channel. Please like, share and subscribe the channel. In this Auxin phytohormone, we are going to learn introduction of plant hormones, introduction of auxin, discovery of auxin, distribution of auxin in plants, mechanism of auxin action, and biochemical or physiological role of auxin with practical applications. The hormones are very important substances which are necessary for proper growth and development in plants. Many external conditions and sufficient amount of food material in the body is essential. But these two factors do not actually control or regulate the growth in plants, but it is done by certain chemical substances which are synthesized in the plants endogenously. Such substances are called plant growth regulators or growth regulated substances. The hormones differentiated from animal hormones, so it is called phytohormones. Phyto means plants, so the hormones which are present in plants are called phytohormones. The chemical substances which are synthesized in the plants to regulate the growth in plants are called plant hormones. These are produced in plant in very low concentration for the effective results to regulate the physiological plant processes. Hormones usually move within the plant from a site of production to the site of action, and this is a very important property of phytohormone. Thiemann suggested the term phytohormones to these chemical substances. Broadly, the plant hormones are classified, and it is also accepted the two major classes: growth promoters, auxin, gibberellins and cytokinins. These are generic groups of hormones which regulate or stimulate the proper growth and development in plants, whereas second one are called growth inhibitors. These are also essential, which are indirectly used for the growth and development, but these are essential in ripening of fruit and initiation flowering and all these things for the senescence of the plants, and these are abscisic acid and ethylene. Auxin is a generic term usually used for a group of compounds characterized by their capacity to induce elongation in shoot cells. These are the first group of hormones discovered. Charles Darwin was among the first scientists to dabble in plant hormone research. In his book, The Power of Movements in Plants, published in 1884, the term auxin is derived from the Greek word auxan. It means to increase or to grow. F.W. Wendt in 1928 was the first person isolated this hormone, but he could not identify it as a growth regulating substance. Auxin is a generic term used for a group of compounds characterized by induced elongation of shoot cells. The group includes both naturally occurring as well as synthetic compounds of auxins are also available nowadays in market. In the discovery of auxin, F.W. Wendt, he has done a very important work from 1928. He has isolated the active substance. To know the chemical nature in 1934, similar active compound was isolated from human urine and identified as a indole 3-acetic acid. In short, it is also called IAA. In the following year, 1935, IAA was isolated from yeast extract. Again in 1946, IAA was isolated from immature corn kernels. After these observations, active growth regulator was isolated from many higher plants, and now it is accepted that IAA, that is indole 3-acetic acid, is the principal naturally occurring auxin in all higher plants. This is the structure of indole 3-acetic acid and the formula is C10H9O2N. In plants, auxin is distributed in all over organs. Now it is accepted that IAA is the principal naturally occurring auxin in all plants from algae to angiosperms. Every young growing plant or bud shows presence of IAA, but its concentration differs in different organs. In angiosperm, the highest auxin concentration is found in stem tip or calyptile tip, and from there translocated in the downward direction. This movement is always polar movement. For proper growth and development as well as for proper physiological function of the organ, certain minimum level of auxin is maintained well in plants. In the mechanism, auxin brings about so many actions within a seconds or minutes or a days in the plant body. The possible mechanism of auxin action involves various events as follows. Auxin IAA is adsorbed to a specific hormone binding site to form an IAA binding site complex. Later on, membrane acidification occurs. IAA increases cell wall plasticity, which easily responds to turgor pressure. The effect of IAA occurs through activation of genes in the plants, which leads to changes in nucleic acids and proteins. Auxin seems to be enhancing both transcription, that is mRNA synthesis, and translation, that is mRNA directed protein synthesis processes. The auxin effect on transcription may be mediated via release of transcription factor from the plasma membrane. This factor also stimulates in vitro RNA polymerase activity. Thus, changes occurring in plasticity of cell leads to growth because of synthesized proteins. The auxins also regulate enzyme cellulose activity during protein synthesis process. The primary physiological effect of auxin on plant growth is cell elongation. It occurs because auxins promote increase in osmotic solutes, decrease in wall pressure, and increase in permeability of cytoplasm to water. The auxin also act through increased enzyme activity of RNA polymerase and involvement of secondary messenger. Recent evidences indicate that auxin increases oxidative phosphorylation in respiration and enhanced oxygen uptake. The growth stimulation might be due to increased energy supply. And the last one part of auxin is biochemical or physiological role of auxin with practical applications. First biochemical role is cell elongation and longitudinal growth. Auxins causes stimulation of the calyptile and stem growth, which varies according to physiological nature of the tissues. This is the primary effect of auxin, which stimulates elongation of cells in shoot, and it is easily observed by Avena curvature test by FW Wendt. Second role is of cell division. Auxin is responsible for initiation and promotion of cell division. Due to rapid multiplication of cells, callus formation takes place, which is very important for healing of wounds in the plant. Callus means undifferentiated mass of parenchymatous cells. Practically, it is used in grafting and tissue culture technique. Callus formation is important and can be achieved by IAA application. In tissue culture, cell division and callus formation is seen because of auxin in the culture medium. Third biochemical role is root formation. The higher concentration of auxin inhibits root elongation but increases the number of lateral roots. This property of auxin is utilized for vegetative propagation of plants by cuttings. In case of many economically important and horticultural plants, auxins like IBA, that is indole butyric acid, 2,4-D, 2,4-dichlorophenoxyacetic acid, and IAA, that is indole 3-acetic acid. These are now commercially used for initiating roots in stem cuttings in many horticultural plants like grapes, roses, etc. The fourth biochemical role is apical dominance. In many vascular plants, intact growing apical bud always suppress the growth of lateral buds, due to which only main stem grows and no lateral branches are produced. If the apical bud is removed, the lateral buds sprout and develop into branches. The influence of apical bud in suppressing the growth of lateral bud is termed as apical dominance. This property of auxin used in agriculture by checking the sprouting of lateral buds on tubers during storage. Potato and other commercially important tubers can be stored for longer duration. Similarly, in case of some horticultural plants, by removing apical buds, branches are allowed to develop for production of more flowers. The next biochemical or physiological role is prevention of abscission layer. Premature fall of leaves, flowers and fruits occur due to formation of a separation layer or abscission layer in the abscission zone, and that zone is at the base of a petiole of leaf or pedicel of flower or peduncle of a fruit. The leaf or fruit fall is due to dissolution of middle lamella and primary wall of the cells in that abscission zone. The development of abscission layers begins due to decrease in auxin concentration below certain minimum level. So premature fruit fall in commercially important plants like apple, pear, citrus is responsible for heavy loss to the cultivators. This loss can be prevented by spraying the dilute solutions of IAA, NAA, 2,4-D like auxins. By spraying plants with low concentration of 2,4-D, defoliation in cabbage and cauliflowers can be prevented. The next biochemical role is effect on flowering. Auxins take indirect part in the physiology of flowering by controlling the vegetative growth, by extending the period of vegetative growth. Through effect of auxin, reproductive growth can be delayed. In some plants, this effect of auxin is synchronization of completion of vegetative growth. This property of auxin is commercially utilized in case of pineapple and sugarcane plants. In pineapple cultivars, use spraying of synthetic auxin like NAA in appropriate concentration. In the appropriate stage, synchronization of flowering can be achieved in all plants in the field, and this makes harvesting easier and less expensive. So cultivators can earn more money from pineapple cultivation. In case of sugarcane, flowering decreases the sugar percentage in the stem. So by spraying the auxin like IAA, vegetative growth can be maintained, which help to maintain sugar percentage in the stem of the sugarcane. This saves the loss of cultivar. Next role is spur formation. Spur means dwarfs which are essential for maximum flowering and maximum fruiting. In apple and pear plants, fruit develops on dwarf branches or spurs. If these terminal buds are treated with NAA, it prevents the formation of elongated branches and increase in the number of short branches or dwarf branches, or these are called spur. This increase in development of more flower buds and fruits. Thus increases the product of commercially importance and profit to the cultivars by the spur formation. Next important role is parthenocarpy and fruit development. After pollination and fertilization, ovule is converted into seed and ovary into fruit. But sometimes formation of fruits occurs without fertilization. These seedless fruits are known as parthenocarpic fruit. Auxin treatment can induce parthenocarpy. In many plants like orange, banana and grapes, natural parthenocarpy can be observed due to internal auxin level. Now, parthenocarpic seedless fruits are developed in many plants which help to improve the agricultural yield. Next biochemical role is eradication of weeds. In agriculture, it is used to destroy various unwanted harmful weed plants. Many dicot weeds are killed by application of synthetic auxins like 2,4-D, 2,4,5-T, 2,4,5 trichlorophenoxyacetic acid with selective activity. Very low concentration of auxins is found effective to control the weeds. Thus, farmers can increase crop by destructing harmful weeds. 10th biochemical role is a prevention of lodging. Many crop plants belonging to Poaceae and Leguminosae tend to lodge. Lodge means fall down due to excessive elongation of stem and softening of cells in the basal internodes of the stem. This reduces the crop yield in many plants. But if auxins like NAA, alpha-niftyl acetamide, is treated to the basal part of the stem, it causes to grow stiff, woody and erect. Thus plants are prevented from lodging and reduction in crop yield is checked. Last one is movements in plants. Auxins are involved in phototrophic and geotrophic movement in plants due to their polar transport and effect on cellular growth. Phototrophic means towards sunlight. Stem is a positive phototrophic organ and it is a negative geotrophic organ, whereas a root is a positive geotrophic organ and negative phototrophic. That means movement of plants or movement of organs towards gravitational force is called a movement of plants, and auxin is responsible for the best phototrophism as well as for geotrophism. If a growing plant is illuminated by unilateral light, then plant bends towards light. This bending or curvature results due to unequal distribution of auxin concentration. All these are biochemical rules. Biochemical role of auxin shows endogenous secretion of hormone, but under certain conditions, low concentration of auxin is synthesized in the plants. Then there is a need of exogenous application or spraying of auxin to the plant. Then it is called practical application of auxin. All these biochemical role we can easily introduce as a practical application in our agricultural field for better growth and development of plant or fruit plant or flower plants. Thank you. Thank you very much for watching this video and also for the subscription of video. Thank you.